So we will talk, me and Annette, about the NDI-6510, which is the result of the COVID Moonshot that was initiated, is it more than 4 years ago now? And also what we will try to show is some of the things which are maybe more hidden in a program going forward, especially towards the preclinical profiling and the results that are generated after you have identified your candidate, after you have achieved your first kind of potent compounds that normally are published. So just to give you a bit of the background, so as you all know, the COVID Moonshot was initiated as a crowdsourcing initiative, very much reliant on fragment screen, and this work, which has been published in Science, led up to the lead compound. Let's see if I can put the pointer up as well. MATPUS E194 DF511. I don't know who came up with acronyms, but it's the compound here, and which had a quite good profile when profiled, and as you can see, or at this stage, there was some alerts identified based on first on the structure, where you can see there were both the potential for racemization of the chiral center, and also built into the compound the presence of the aminoacequinoline as a genotoxic motif, and mitigating these risks was an early kind of goal for the lead optimization program. And also the data indicated that the compound were cleared quickly across all species tested. So this was also something that needed to be addressed early on in the program. But, sorry, I just need to throw my dog away. So at an early stage. So the important, and if we can't lose the Ames activity, then you don't have a program, because this is something that will kill a program at an early stage when you do your preclinical genotox studies, and it's a regulatory hurdle that is very difficult to overcome if you have a Ames activity built into your compounds. And very early profiling of the compound showed that you have a positive Ames activity in the Ames strains tested. So you can see you're testing always, when you're testing Ames, you're testing under non-metabolizing conditions and metabolizing conditions, and there's no kind of dose response in this. So either it's active or it's not active. If it's active, then you need to sort a problem, and the only way to sort a problem is to change your structure. So this is the work that was done as an upfront effort to allow the progression of the compounds. And you can see some of the initial studies when you look on the aminoacequinolines with different substitutions in the 7th position. You can see that the compounds tested here, they were all positive in at least one of the Ames strains tested, as is the full compound, and this is probably due to that in your assay you are cleaving off the aniline moiety that can be further kind of processed to genotoxic species. So this was the first step. And to address this problem, there was a few ideas on how to address this. One is can we find a Ames-free isoquinoline replacement? Second is, can we do blocking of the position that is oxidized by positions on the isoquinoline, and with any substitution in the 6th position? We already showed that in the 7th position this is not the case, that if you introduce substitution, would that change the genotoxic potential? And also, could you lock the aniline to prevent the release of the 3-amino isoquinoline? Could that be done in one way or another? So I will go through what was done in this way. So the first step was, could we identify Ames-free isoquinoline that retains decent SARS-CoV-2 Mpro potency? And as you can see from this slide, with all the example tested here, you lose quite a lot of, or you lose a lot of the Mpro activity in the compound. And the only case where you saw some retention of activity was when you used this derivative here. But of course, the risk here is that you get re-aromatization of the unsaturated ring back into the aniline. So this was deemed as an approach not suitable for further exploration. We also in the work tested with a 6 substitution, and that also led to the decision not to progress that approach because of the risk for aniline formulation and formation in the synthesis. The approach that proved successful in the end was to lock the aniline into a chemical structure or a motif that don't release or don't have the possibility to release a free aniline. And this was done to spiral cyclization. And you can see some of the spiral systems that were tried and how they position themselves in the X-ray structures of the SARS-CoV-2 main protease. And you can see that they very well mimic the non-cyclized compound. And if you look on the IC50 values, you can see that they are fairly well in line with each other within threefold. So they're all kind of possible alternatives. But when looking a bit more deeply into this, you can see that when built into the system and trying testing across several different inhibitors, you can see that for some of the systems you have a more substantial drop-off than for others. And if you go through them one by one, so you have the hidantine where you see about 4-fold drop-off. The same is for the imide ring. We have a carbon instead of nitrogen and 2 oxygens. And also the same is seen for the lactam system. But for the lactam system, if you choose another different kind of side chain on the tetrahydroisoquinoline moiety, then you lose much less of the potency. So there seems to be an opening in terms of retaining potency and creating a system which will not have the possibility to release a free aniline. And also just to mention that theoretically you can release the amino isoquinoline from both the hidantine and the imide-based system. So in the end, it was the lactam were prioritized over other alternatives. Yeah. And moving forward, you can see that when testing lactam-based compounds, these are now don't show any activity in the Ames strain, and this has been the case for all subsequent compounds from this class tested in the Ames assay. So this seems to be a good approach to avoid release of genotoxic metabolites. So this problem now solved. So moving to the next big problem, which were the metabolic liability built into the original lead structure. And what was tried here by Ed and the team was to go through the different, blocking the different possibilities for metabolic processing. And I should say that in the studies that were done, we were using cassette dosing in rats. So we dosed in vivo a set up to 5 compounds and then looked on the clearance of the compound. So, and going through the different alternatives. So aromatic oxidation is one possibility. You can also think about N-oxidation of the nitrogen, benzylic oxidation, and side chain cleavage were investigated. But first look into what is the consequences on the metabolism when doing the spirocyclization. And from this kind of match pair plot, you can see that on average, when doing the spirolactamization, you lose around threefold of your potency, but you also gain some stability. So it's a kind of fairly good conversion, but it doesn't take you the whole way. Looking on the aromatic oxidation, you can see here you don't really see a benefit neither on the potency or on the metabolics, increasing the metabolic stability. So this is probably not a path forward for further to fix the metabolic clearance. The third approach, which actually impacted quite a lot, is to block the benzylic position on the tetrahydroisoquinoline moiety by introducing a carbonyl group. And here you can see in the average impact on potency, it's positive, almost twofold, and you see a substantial decrease in the rat clearance. So that's a very good modification on average. We also did investigate blocking the side chain cleavage on the tetrahydroisoquinoline, and in this case, there was a clear confirmation of preference for when blocking the methylene position. So if you have a cyclopropyl, you can see that the potency is much better compared to if you put the dimethyl, and this can be explained by the conformational preference of the side chain. So in the end, the compound without any substitution, the cyclopropyl, were the kind of chosen compounds for further investigations. And yeah, just to summarize, so the kind of end result of all this SAR exploration, all the efforts, was the identification of the 3 lead compounds or late lead compounds, the NDI 65, 12, 15, and 10. And based on the mainly on the metabolic properties of the compound, the NDI 6510 was selected as the primary candidate for further investigation, although that we did do a lot of work on 6512 and 6515 to see if we could qualify the compounds. And you can also see that for all these compounds when tested in the Ames assay, they are still negative. So these the 2 kind of big problems of the lead was fixed during the ELO program. So now over to Annette to discuss on the in vivo profiling.
So we took these 3 lead candidates ahead and profiled them against the virus initially. And what you can see here are the results from a CPE-based assay, so a viral assay that is based on cytopathic effects, where you basically look at rescue from killing of cells due to the virus. And you can see here that all of these compounds sit in a kind of similar potency range to clinically approved inhibitors, ensitrivir and nirmatrelvir. And when you look across viral strains, so we call these variants of concern against SARS-CoV-2, we do see potent inhibition against all the different SARS-CoV-2 variants. And here we included beta strains, Delta, Omicron, and various Omicron strains. However, the spectrum doesn't extend beyond SARS and SARS-CoV-2 activity. So we do still see activity against SARS-1 across the different compounds, but not against, for example, 229E. We also tested OC43 and MERS, and our compounds are not active against other non-SARS or SARS-CoV-2 coronaviruses. At this time, we felt that the spectrum was sufficient to move the compound ahead into further evaluation. So next slide, please, Peter. And we wanted to see whether the compounds would be efficacious in vivo. So we looked further into how we would assess this, and the kind of model that we chose was a mouse-adapted strain of SARS-CoV-2. However, when we looked at the PK of our compounds, and you can see that here on the blue line, our compounds, despite the quite significant improvements on the rodent metabolism, were still rapidly cleared in animals. So if you give this compound to a mouse at a dose of 50 mg/kg, you do get an exposure of about 4 or 5 hours over 3 times EC90, which is the green line here, but not beyond that. And what we would like to see for BID dosing, so twice a day, is a exposure of about 12 hours over EC90. So we adopted a strategy of using a CYP3A4 inhibitor, which is called AViDD. And when we co-dosed that, we did achieve, if giving higher doses of our compound of 15, 50 and 150 mg/kg exposures over the 3 times EC90 to about 12 hours. Because this is kind of linked to co-dosing an inhibitor, we also explored an alternative strategy, which is looking at a humanized mouse model. And this work was performed in collaboration with the University of Dundee, where we worked with Kenny McLeod and Kevin Reed to access the humanized mouse model, which they called ATAM. It's recently published in PNAS. And in this mouse model, at doses of 50 mg/kg and 150 mg/kg, we do see, again, an increase in exposure compared to wild type, and that's very much linked to them replacing all the rodent CYPs with human CYPs, and also PXR and a couple of other metabolites and induction targets are replaced by the humanized version. So we then went on and used these 2 models to dose our compounds to mice. So that's on the next slide. And the aim here was really that our compound showed a very good human metabolic profile, but not rodent. So we wanted to overcome the rodent-specific metabolism to kind of show an in vivo effect. So this is the animal efficacy that we ended up running. On the left-hand side, you can see that we co-dosed our compound with AViDD BID. So the AViDD was given once daily and our compound twice daily. And then on the right-hand side, you can see the humanized mouse model that we ran, where we just dosed our compound BID. And then at the end, we assessed the mouse weight and lung viral titers and also the histopathology. And what you can see on the left-hand side, looking at the 3 doses of our compound, we didn't see a substantive effect on weight loss that is caused by infection for our compound, whereas for nematrivir, you do see some recovery of weight to the uninfected state. And then on the right-hand side, the picture is a little bit more diverse. So here you can see that we did impact on weight, but this actually exceeded the uninfected weight loss, and also again above vehicle, which is again in the range of nematrivir, which is shown in orange. But you also do see that at the lower dose, we don't recover that weight loss. And at the highest dose, which was 300 mg/kg, we do see a weight loss that actually exceeds the vehicle. So on the weight loss, we basically didn't see a clear signal that our compound was doing anything. Next slide, please. When we then looked into viral loads, so lung viral titers in the lung, On the left-hand side, you can see the results for the ABT co-dosing model. And there we didn't see any effect on viral loads. And what we think now is that the once-a-day ABT dosing wasn't sufficient to exceed EC90s consistently, or 3 times EC90 consistently, which we think is the cutoff that you have to have your concentrations above to see a readout of viral load decrease. However, if you look on the right-hand side, you can see that even at low doses of 50 mg per kg, we did see a significant reduction in viral loads, and that is more pronounced for the higher doses of 150 and 300 mg per kg, but much exceeded by the very high dose of nirmatrelvir that we used here as a positive control, where you basically have undetectable viral loads. So we are very confident that this experiment worked very well. However, we didn't see the same log reduction for our compound that we see to nirmatrelvir. One has to say here that nirmatrelvir was dosed at higher doses that were taken into the clinic if you compare that. On the right-hand side, if you look at histopathology scores that we performed for this experiment, we also did see a significant reduction in histopathology scores, similar to what is seen for the positive control. Next slide, please. Based on that data, we went ahead and tried to identify the maximum tolerated dose in exploratory toxicology studies. So we scaled the compound up to 500 grams and then dosed the compound to rat over 3 days. So, and then at the end, so actually on day 1 and day 3, we looked at the plasma levels for doses of 100, 300, and 1,000 mg per kg. And the compound was well tolerated for these high doses. What you can see on the left-hand side for rat is that you can see excellent dose proportional exposures on day 1. However, when we look at dosings or at exposure levels on day 3, and the AUC is plotted here, we do see a significant reduction of exposures on day 3 in comparison to day 1. So based on this data, we concluded that we saw significant induction in rat. Our first thought was that we should investigate whether we see the same kind of effect in a secondary rodent species, because that would give us an alternative rodent species for the MTD studies for preclinical development. However, when we ran an explorative PK study in male mice, looking at 5 days of exposure, we do see the identical effect where we basically lose the exposure almost completely after multiple doses. This effect wasn't observed in dog, where we did see increased exposures and actually supraproportional exposures on day 7. So there seems to be a kind of rodent-specific effect. And that led us to follow that we wouldn't be able to reach sufficient exposures above the predicted human dose for DNDi 6510. We did look a little bit into the proposed mechanism for this induction. We initially, in our initial PXR assays, we didn't see a signal for our compounds, but we then went back and did a more comprehensive profile and looking in more detail into the different nuclear hormone receptors across 4 different species: mouse, rat, dog, and human. We did see significant PXR-linked induction. And when we followed this up in a cryopreserved hepatocyte assay, where you basically take human hepatocytes. We did look at both mRNA levels and catalytic activity. We did see that when treated with DNDi-6510, we got significant induction both on mRNA level and also on catalytic activity that you can see in the bottom right corner. However, we didn't see any effect on any of the other investigated CYP enzymes. So this seems to be a PXR-linked CYP3/4A specific effect that is leading to the phenotypic lack of exposure on multiple dosing. Next slide.
That's me again. So in parallel to the investigation and profiling of compounds, we did invest a lot of efforts into scaling up and providing a synthetic route that could produce kilos of the DNDi-6510 for both for the toxicology studies and also as a kind of material for the first human studies. So this was done on a GMP manufacturing level. So just a few words on the synthetic route. I won't go through any details. So there are, as you see there, it's a very steppy route. There are 12 synthetic steps in the route, in this route, and also that you have a mid resolution mid stage where you step 6, where you lose 50% of your material in the resolution. So, and this is dependent on that you have a chiral center, which is actually a 4-carbon chiral center, so it's quite unusual. And this leads to an overall low yield in the process. There's a number of steps, and also that you waste 50% of your mass halfway through the synthesis. And it was not uncomplicated to develop this route, so it took quite some time, much longer than expected, to get a route that actually could produce the material. And as you can see on the scales that we were operating, so the key intermediate 7, that was produced in about 80 kilos, so it's big scale. And then we performed a salt resolution in a 30 kilo scale. It worked very well, but still a large scale. And then one of the more tricky steps that we needed to work a lot with was the Ullmann coupling, where you attach the isoquinoline to the intermediate 10, where the sensitivity of the reaction, for example, presence of impurities or small amounts of oxygen could kind of reduce the yield significantly. So I think we were for a long period in a range of 40 to 80% yield in this step, and to get this to work constantly at high yield was a significant effort. But as you can see, finally we performed this step, the Ullmann coupling, at producing 16 kilos of material at a 95% yield. So that was a really tremendous piece of work by the Samaras team who did the work. So in the end, we managed to produce more than expected, 10.6 kilo of material for the GM suited for downstream studies. To say, just to kind of say that this wouldn't be the route that you could use for manufacturing of commercial supply. So there is, in the downstream development, we did plan for radical redesign of the synthetic route that would lead to kind of lower costs of the API in the end. Also, just during the work, we kind of did absolute confirmation of the stereochemistry by X-ray crystallography, and for the key intermediate S7, and this, since it's a 4-carbon chiral center, this won't change throughout the synthesis. We were 100% certain about the stereochemistry. A final word before leaving over to Annette for the conclusions is that with the material in hand, we did also investigate the possibility to to generate material suited for kind of formulation work. So we did do a salt screen, which didn't produce any kind of salts that offered an advantage over using the free base form. And we did do, on the free base form, we did do a polymorph screen that identified one stable anhydrous polymorph. We call it form 11. However, this form in aqueous media converted into a thermodynamically more stable monohydrate. Unfortunately, this monohydrate has a lower solubility compared to the anhydrous form. So this requested, and you saw that we used corn oil formulation. So we needed to have a formulation for our efficacy studies and also for our PK studies, where we were certain about the form of the material in the suspension. So we didn't want to see any form conversion during in our administration vehicles over time, for example. And also a consequence of this is that we, in the downstream work, if we should have progressed, this would have led to an increased investment in development of clinical formulation. Good. Over to you, Annette, for the final words.
Okay, so this is our last slide. Sorry, we ran over time. So in summary, we identified a potent SARS-CoV-2 Mpro inhibitor that ameliorated previously or identified AIMS and metabolic liabilities. We demonstrated in vitro and in vivo efficacy across a range of SARS-CoV-2 variants, but not MERS or other coronaviruses. In MTD assays, to very high doses of 1,000 mg per kg, we detected significant PXR-linked CYP3A4 induction, particularly in rodents. And this ultimately led to the preclinical development of DNDi-6510 to be discontinued based on the consideration that we wouldn't reach sufficient toxicology windows as well as a sufficient exposure for genotoxicology studies later on. We identified a workable synthesis route and characterized solid state and polymorphs. And what we have done over the last couple of months is worked on a data release where we are going to disclose all the preclinical data as well as late lead-up data through Kemble and working on an open CDD Vault release as well. And you can find all this data in a preprint that was actually published yesterday. So it's a very timely open science forum. And I think the last slide just concludes with some acknowledgements to all the previous Moonshot partners, but also the partners of the preclinical development team.